I'm Jason Coe from the University of Maryland, Baltimore County, and today I'd like to talk to you a little bit about my research modeling sorting, intercalation, and involution tissue behaviors due to regulated cell adhesion. So as a short introduction, dynamic changes in cell adhesion are critical to multicellular development, as seen in normal embryogenesis, like the development of the germ layers involves differences in adhesion that separate those layers from each other, as well as cancer metastasis, right? So you have a tumor, and then cells have to undergo changes in adhesion to break off from that tumor and then move elsewhere in the body and metastasize. However, it's unclear exactly how regulation of adhesion drives developmental dynamics. So we developed a novel modeling approach where we model both cells and the adhesion molecules they express, and in this way, we can capture regulated adhesion dynamics. So here is our mathematical model. So we have a variable u that models the cells and a collection of variables ci that model cell adhesion molecules, or CAMs. And you'll notice that they are subject to the same velocity v. This is because CAMs are membrane-bound proteins, and so as cells move through space, they carry the CAMs with them. Similarly, as cells proliferate, that results in increased CAMs. CAMs can also be produced in accordance with the action of certain morphogens, which are carried by the tissue, but also diffuse throughout the tissue. And morphogens can also lead to the production of other morphogens. And so the velocity by which cells move has two components, dispersion and adhesion. And adhesion functions on pairs of cells within some radius r of each other. The strength of that adhesion depends on two things. There's which CAMs are interacting with each other, which is determined by this constant, a sub ij, as well as quantitatively how much CAMs present at the two pairs of points that are being pulled towards each other. There's also a crowding capacity function h that makes it so that we can't have all points in space collapse onto a single point due to adhesion, which is mathematically possible but physically infeasible. And here's an example of a matrix that encodes some adhesion constants for a system of two CAMs, red and green. And then we can see cell density and CAM concentration distributed in space here. And so we can see that these two things are co-localized. Scenarios that we can model with this approach is cell sorting. And so in classical experiments, it's been demonstrated that cells with greater adhesive properties move towards the center of aggregates as they sort, as opposed to cells with lower adhesiveness get pushed towards the outside of the aggregate. And we see that if we encode high adhesive strength in the red population and low adhesive strength in the green population, that indeed we do get this sorting as in the experimental data. It's also necessary to specify an intermediate value for the adhesion between red and green. And we see that as we vary these adhesion parameters, we get a variety of different dynamics. And we can demonstrate that these dynamics are a result of the adhesion parameters and not on the initial state. Explain intercalation dynamics. Right, so we see here on the right half that we have two different populations of cells, red and green, and the red population is expressing nectin, or is transfected with nectin-1, and the green population is transfected with nectin-3. So as these cells proliferate, in both the experimental case and in our simulation, when they meet in the middle, we get this intercalating, the knitting of the cells together. And this is due to their adhesive properties and not due to simply proliferation of cells, because we can see when all cells, left and right, express the same nectin-1, and thus have the same adhesive properties. As they proliferate, they get contact inhibition, and this is the same as in the experimental case, and these constants follow from empirical data. Simulation of zebrafish gastrulation, we have a diffusing morphogen nodal, which leads to the upregulation of a CAM and cadherin, and we specify some adhesion strengths in this matrix as before, and this causes cells to involute around the germ ring margin and move towards the animal pole over time, as seen here. And this is particularly interesting because nodal is only expressed in the EYSL region of the yolk and only diffuses into a very small region of the blastoderm, close to the germ ring margin here. And even so, as cells continually move out of that region due to adhesion and new cells move in and are constant, so you have constantly new cells exposed to nodal and thus upregulating cadherin, this is able to drive a continuous motion of cells upward towards the animal pole, which is fairly interesting. And so to conclude, we presented a novel mathematical model of dynamic cell adhesion that can explain a variety of developmental cellular dynamics. And I'd like to thank NSF for funding, as well as UMBC and my mentor, Dr. Lobo, for their support. Thank you.